Welcome to AP Biology. Today I want to talk about what happens after glycolysis. So in glycolysis, glucose is broken in half to two pyruvic acids. So what happens to that pyruvic acid? The ATP that's made, that can just go ahead and be used, right? So anytime you need to contract a muscle or move sodium out of a neuron, anytime you need ATP, you can just use that. But what's going to happen to this NADH? So that's what I want to talk about next. So the ATP can be used anytime a cell needs energy. But what happens to the pyruvic acid and the NADH? Well, that depends on whether we have oxygen present or not. So some organisms can do a process called aerobic cellular respiration. That's what we do. And they can break down the pyruvic acid and use the NADH. Some organisms can't do that, and they do fermentation instead. We actually can do one form of fermentation and aerobic cellular respiration. Other organisms do anaerobic cellular respiration. Both of these processes, fermentation and anaerobic cellular respiration, for both of those, you need no oxygen. For this one, aerobic, you need oxygen. So what happens to the half of glucose and to the NADH depends on whether you have oxygen or not. If you've got oxygen, maybe you'll do aerobic cellular respiration. If you don't, maybe you'll do fermentation or some type of anaerobic cellular respiration. The versatility of an organism really depends on their enzymes. So some organisms can do more than one process, and some organisms can only do one of these processes. But we're going to look mainly in this unit, we're going to look at aerobic cellular respiration, this one. But we'll just very briefly talk about the other two, the anaerobic types. And then I have this picture of a lightning bolt just to remind you that NADH is carrying these high energy electrons, and we're going to talk about what happens to those. All right, so since cells don't have a lot of NAD, the NADH has to be oxidized to make more NAD. So what does that mean? Okay, so NAD is the same molecule as NADH, except this one has some extra electrons, right? So this one can take electrons, a couple of electrons and hydrogen, right? It can pick them up and become this. So NAD plus and NADH are really the same molecule. It's just that one has the extra electrons and one doesn't. So if you just do glycolysis, that's this step, glycolysis. Let's say you just do glycolysis and then after that you don't do anything. You're going to build up a lot of this and you're going to have very little of this. You're going to have less of that. And you're going to keep using what you've got and eventually you're going to have loads and loads of this. So lots of NADH and you're going to have no NAD plus after a while, and that's not good because then glycolysis can't keep working, so you can't keep making ATP because remember, the process of glycolysis also makes ATP. So that's not good. So somehow you've got to find a way to get rid of those electrons and the hydrogen and get back to NAD+, right? And so the way that you do that depends on whether you have oxygen present or no oxygen present. So if you have no oxygen present, you're going to do something called fermentation or anaerobic cellular respiration. And if you have oxygen present, you'll do this other thing called aerobic cellular respiration. But either way, you've got to ensure that you're still making more of this so that you can do more glycolysis. So this ensures that glycolysis is going to continue. Because this guy needs to do something. He's this one, right? Well, if he has his electrons. So anyway, at some point, he's going to have to lose his electrons and hydrogens. So how will that happen? Okay, so pyruvate, remember pyruvate is a half of glucose, right? So here I've got some glucose, and if you break it in half in glycolysis, you've got 3 carbon compounds, you've got 2 of them, 3 carbons, you've got some oxygens and some hydrogens. That's pyruvate. So pyruvate is the branching point. Everybody does glycolysis. All the bacteria in the world, all the archaea, the fungi, the plants, the animals, the protists, everybody does glycolysis. But once you have pyruvate, what you're going to do next depends on the enzymes that you have and depends on whether there's oxygen or not. So if there's no oxygen, you can do fermentation, or you could do anaerobic cellular respiration. Show you that here. If there's no oxygen, you're going to do fermentation, which is an anaerobic process. We're going to come back and talk about that toward the end of this unit. There's something else, though, that I should have put in here. There's also anaerobic cellular respiration. So anaerobic cellular respiration is a little different from fermentation, and we'll talk about the differences later on. If you have oxygen, though, if you're a eukaryote in your mitochondria, you can do the Krebs cycle and the electron transport. You can do aerobic cellular respiration. So that all happens in the mitochondria right here. This is sort of the same picture, but in a cell. So glycolysis just happens in the cytoplasm. Fermentation just happens in the cytoplasm. Same thing with anaerobic cellular respiration. Well, it would, because it would be bacteria. Anyway, here's a mitochondrion, and aerobic cellular respiration happens in the mitochondria. So we're going to spend most of our unit on aerobic cellular respiration, but just a heads up, we are going to come back and talk about fermentation and anaerobic cellular respiration. Remember, both of those are no oxygen. Okay, so we're going to look at what happens to glucose when oxygen is present, aerobic cellular respiration. And all of these next steps are going to happen in the mitochondria if you're a eukaryote. That doesn't mean prokaryotes don't do this. They do. So if you're a prokaryote, you would say in the cell, whoops, just in the cell and the cell membrane. For any prokaryotes that do this. So we're going to spend most of our time talking about eukaryotes and mitochondria, but I'll come back at the end and we'll talk about bacteria too. Prokaryotes, remember, no nucleus, no mitochondria. They can still do this process, a lot of them can, but they wouldn't do it in a mitochondria because, of course, they don't have any internal membranes, and boy, this mitochondrion sure is membranous, right? Okay, so in the presence of oxygen, the pyruvate, the half glucose, enters a mitochondrion, if you're a eukaryote, and the oxidation of glucose is completed. What does that mean? So you've got a half of glucose now, you've got pyruvate, and it's got some O's on it and the H's, which I'm not going to draw, but the oxidation means that you're going to be ripping off electrons, right? Probably some hydrogens too, and it's completed, meaning you're going to end up just with carbon dioxide. So with these 3 carbons attached to their O's and their H's, you're going to have no more H's on here, just a carbon. Okay, so that's what I mean by completed. You break it down, you break that half glucose down completely. So in these steps, we're going to cut the fuel up even more. We'll chop it into pieces that are only 1 carbon big, meaning carbon dioxide. It's now inorganic. And remove every last hydrogen from it and take a bunch of electrons off of it too. Those electrons are going to go to NADH, and we'll talk about that like a long time from now. So it's like this long story, and you've got to wait till the very end to find out what happens to NADH. All right, so this is the step right before the Krebs cycle, right before we get into the citric acid or the Krebs cycle. So before the citric acid cycle, that is actually the same thing as the Krebs cycle, by the way. Before the Krebs cycle, it's supposed to be a K. The cycle was named for one of the molecules in it, citric acid, or it was named for the guy who figured it out, Dr. Krebs. I don't know his first name. Anyway, it's the same thing, and so I'm sorry that there's extra vocab words for you to learn. It's kind of a bummer, huh? So anyway, before it can begin, this half of glucose, this pyruvate, has to be converted to something called acetyl coenzyme A, but everyone just calls it acetyl-CoA for short. So at the end of glycolysis, we've broken glucose in half, and we have 2 pyruvates. Here's what pyruvate looks like. Carbon, carbon, carbon. There's 3 carbons. You take off this part, and you make carbon dioxide. And then you rip off some hydrogen and some electrons, and you make some NADH, 2 of them, well, because there's 2 pyruvates. And then you put on coenzyme A, and that's this part of it. And so you make this thing called acetyl-CoA. So in this pre-Krebs step, that's what I'm going to call this, pre-Krebs, because we haven't actually gotten to the Krebs cycle yet, pre-Krebs step. In this pre-Krebs step, we've already made some CO2. So remember, before glycolysis, we started out with glucose, which had 6 carbons, C6H12O6, right? And then through glycolysis, we ended up with 2 of these 3 carbon compounds, 1, 2, 3 carbons, right? That equals a 6. And already in the pre-Krebs step, we've made some carbon dioxide. I show just 1 made here, but you're actually going to do this with 2 pyruvates, so you'll be making 2 CO2s. So that means you've only got 4 more CO2s to pull off eventually. All right, so anyway, you've also ripped out some electrons and made some NADH. So now we have even more NADH. Remember I said we have to remake the NAD eventually? Right now you're making even more NADH. And you've got this thing called acetyl-CoA, which is going to go into the Krebs cycle, which is our next step, Krebs or citric acid cycle. What else do I want to say? This step is carried out by a multi-enzyme complex that catalyzes 3 reactions. Step 1 is to take out the CO2. Step 2 is to rip off some electrons and hydrogen. And step 3 is to put coenzyme A on it. So there's not one enzyme involved in cellular respiration. There are several enzymes involved just in this one tiny little step. All right, and that is where I'm going to end this video. Bye.